In this presentation, we'll look at different life cycles that organisms have and generally how diploid organisms form reproductive cells, or gametes. We'll also look at why biologists think that sexual reproduction is so common. In the presentation on mitosis, we looked briefly at the human life cycle. In fact, we should expand that a little bit in order to describe the difference between gametes and somatic cells. Let's start on the right-hand side with the fertilization of an egg with sperm. The results of that fertilization is a zygote, and that zygote is a single cell with 2 copies of each chromosome. Having 2 copies is what we call diploid. The individual grows and divides many times by mitosis. These divisions result in many cells, and as development progresses, these cells become specialized. Despite the specialization of the cells, they retain the same genetic information and are genetically identical to the original diploid zygote. All of this mitotic cell division produces what we call somatic cells. There are, however, a small number of cells that divide by meiosis to produce eggs and sperm. In contrast to mitosis, meiosis does not produce cells that are identical. Instead, it produces cells that have exactly half the amount of DNA compared to the mother cells. These are called haploid cells. We can look at the ploidy number of the different cells during the life cycle. Once fertilization happens, the zygote is a diploid with 2 copies of each chromosome. These 2 copies are represented by 2n, and as humans, we have 23 pairs of chromosomes. In all, we have 46 chromosomes, and so we designate this as 2n equals 46. This signifies that we're both diploid and that we have a total of 46 chromosomes, or 23 pairs. The products of meiosis, eggs and sperm, are haploid, and we designate this as n equals 23, signifying that they are haploid with only 23 chromosomes. Remember that we previously looked at a karyotype and saw the 23 pairs of chromosomes for humans. Remember that a karyotype is made during mitosis, and so at that time the sister chromatids are still attached together, as shown here. Moreover, humans are diploid, so we carry 2 copies of each chromosome. These are called homologous pairs and, remember, have the same genes in the same location, but they aren't identical because one came from the father and one came from the mother. So we're all probably pretty familiar with the human sexual life cycle as we looked at it a moment ago, and the same system is common for most animals. Diploid zygotes divide by mitosis to form diploid multicellular adults, and in specialized cells undergo meiosis to form haploid gametes. Haploid gametes fuse in fertilization to then form a diploid zygote again. In plants, the system's a little bit different because plants have a multicellular haploid phase. Again, let's start with fertilization. The 2 haploid gametes fertilize one another to become a diploid zygote. The zygote grows by mitosis to become a multicellular diploid that is called a sporophyte. Some cells of the sporophytes undergo meiosis to form spores, and these spores are haploid. The main difference with the animal cycle is that these spores then grow up into a multicellular haploid organism with all the cell divisions again happening by mitosis. Eventually, haploid gametes are produced, and these fertilize each other to produce zygotes again. In most fungi, the system is different again, with unicellular diploid zygotes undergoing meiosis to form haploid organisms that may or may not, depending on the species, divide by mitosis to become a larger multicellular organism. Eventually, gametes are produced by mitosis and these gametes fertilize one another to again produce a unicellular zygote. In a later class, you'll learn more details about these other cycles. For now, notice that meiosis always reduces diploid to haploid. Mitosis, on the other hand, is a copying mechanism that makes multicellular diploids out of unicellular diploid cells. Mitosis also makes multicellular haploid cells out of unicellular haploid organisms. So meiosis is always reductive. It reduces the genetic composition by half, but it doesn't always produce gametes, as you can see in the example of the plants. Since one of the most important things about mitosis and meiosis is tracking the chromosomes, let's sure we get that straight. In all of these pictures from your textbook, the paternal chromosomes are shown in blue and the maternal chromosomes are shown in red. That means that anything in red was inherited from the individual's mother and anything in blue from the father. In this organism, the diploid number is 6. That is, 2n equals 6. And there are 6 chromosomes in total with 3 pairs. As in the cell shown here, meiosis is about to begin. We know that because S phase is complete and the chromosomes have been duplicated with sister chromatids lying alongside each other. We can identify the homologous pairs of chromosomes by their size. Remember that homologs have the same genes in the same order, but are not identical. Because they have the same genes, they're the same length and can be identified by length. In humans, the longest chromosome is called chromosome 1. The second longest is chromosome 2, the third longest is 3, etc. At least, that was the idea, but some chromosomes were misidentified by length when the numbering system was developed, and so this isn't strictly true. If you've seen diagrams of meiosis, you may think, wow, that's pretty complicated. But in fact, if you understand mitosis well, meiosis will be easy for you. Let's start with an overview of how meiosis works, in general, to accomplish the formation of gametes. This cell has a diploid number of 1. That is, this organism only has a single chromosome, but it is diploid, with a blue chromosome coming from the father and a red chromosome inherited from the mother. During S phase, the chromosomes are duplicated to create sister chromatids. Meiosis occurs in 2 phases. During meiosis 1, the homologous chromosomes are separated and the cells divide. This results in one cell with the paternal chromosome and one cell with the maternal chromosome. In the second phase of meiosis, or meiosis 2, the sister chromatids separate to form 4 haploid gametes. During meiosis, many of the same things happen in the cell as in mitosis. For example, the nuclear envelope breaks down in prophase and the spindle apparatus appears. For my classes, I'm most interested in what happens to the chromosomes themselves. During prophase, the chromosomes condense and homologous pairs line up with one another. This lining up of chromosomes is one of the major differences between meiosis and mitosis. In meiosis, each chromosome is independent from one another, but in meiosis, the homologous pairs line up with each other so that chromosome 1 from the mother lines up with chromosome 1 from the father. Chromosome 10 from the mother lines up with chromosome 10 from the father. Even the X and Y chromosomes, which we haven't learned about yet, line up with each other. We'll look in more detail at this lining up of chromosomes and a process called crossing over, which affects chromosomes at this stage. During prophase, the centrosomes divide and migrate to the ends of the cell, and microtubules extend from them. During metaphase, the spindle fibers finish attaching to the kinetochore of each chromosome. One set of spindle fibers is attached to each member of a homologous pair. As with mitosis, the chromosomes migrate toward the metaphase plate and wait there until anaphase begins. This is another major difference between meiosis and mitosis. In mitosis, because the homologous chromosomes aren't lined up with each other, each chromosome is independent. However, in meiosis, homologous pairs are still attached, and so when they line up at the metaphase plate, the homologous pairs line up together in what is called a tetrad. At the beginning of anaphase, the proteins that hold the sister chromatids together, cohesins, break down, and this allows the homologous chromosomes to separate. You'll see why the loosening of ties between sister chromatids allows homologs to separate when we talk about crossing over. As the motor proteins on the kinetochore move along the microtubules, homologous pairs are separated, but each homolog drags the sister chromatid along with it in pairs. In telophase, the nuclear envelope may reform in some species, and cytokinesis again divides the cytoplasm. At the end of meiosis I, there are two cells that are genetically distinct from one another. They're distinct because each has only one homolog from the homologous pairs. During meiosis II, the same processes are repeated. Note that there is no DNA synthesis between meiosis I and meiosis II. Cells start meiosis II immediately after meiosis I. In prophase II, the nuclear envelope again breaks down if it reformed, and the spindle apparatus again forms and separates. In metaphase, the kinetochore microtubules again pull the chromosomes onto the metaphase plate. At this stage, notice that there are no homologous pairs in either of the cells. This is much more like mitosis. During anaphase, the last of the proteins holding the sister chromatids together break down and pulling along the microtubules results in the sister chromatids being separated and migrating to the poles of the cells. Telophase reforms the nuclear envelope, and the spindle apparatus disintegrates, and finally cytokinesis divides the cells. Note again that the product of meiosis is a single cell and is the production of not two identical cells, as in mitosis, but 4 unique haploid cells. Let's take a little closer look at what happens during prophase I, when the homologous chromosomes line up against each other. Remember that after DNA synthesis during S phase, sister chromatids are held together by proteins called cohesins. Cohesins help keep the sister chromatids aligned, and homologous chromosomes line up in parallel to each other, with each gene along the length being matched up. The homologs are then held in place by a protein complex called the synaptonemal complex. When the chromosomes are fully aligned and the synaptonemal complex is formed, a break forms in each of the chromosomes, and ends of the chromosomes are swapped. This results in new chromosomes that are combinations of the maternal and paternal chromosomes. Remember that the sister chromatids are connected to each other with cohesins. If we look at the sister chromatids on the red strand, we see that the top one here is continuous and has no breaks in it. The lower one, however, has a break right in the middle, and again near the end here. The red chromatids remain stuck together with cohesins, but the actual chromosome is rejoined to the blue chromosome at both of these chiasmata. As the synaptonemal complex disintegrates, the chromosomes move apart from each other and the chiasmata become visible. Notice that in this diagram that all of the crossing over of the red chromosome occurs on one sister chromatid. Both of the blue chromatids are involved in crossing over, one at the first chiasmata and the other sister chromatid at the other chiasma. Human chromosomes typically cross over an average of 1.5 times per chromosome. We'll learn in a later lesson how to estimate the distance between genes along a chromosome as a result of the possibility of crossing over between those genes. For now, you can see that the closer together two genes are along the chromosome, the less likely there is to be crossing over between them, even though crossing over is very likely along the whole length of the chromosome. Let's look at two ways in which meiosis can generate diversity. The first mechanism is through crossing over. In this example, the cell only has a single chromosome that's been duplicated and is now in meiosis. Crossing over between the homologs generates some molecules that are recombinant. That is, they're combinations of the maternal and paternal chromosomes. As we track those chromosomes through meiosis, we see that first, the homologous chromosomes separate, and second, in anaphase 2, the sister chromatids separate. Because of crossing over, the sister chromatids are no longer identical to one another. The 4 haploid daughter cells are each unique with respect to their genetic makeup. Although all of the genes are present from this chromosome in each gamete, the exact copies of them are in different combinations. In humans, there are at least one recombination event for each homologous chromosome pair, resulting in 4 different gametes every single time for every single chromosome. There's a second important way that meiosis can generate diversity, and this is known as independent assortment. Look at the meiosis happening on the left. In this case, just by chance, both paternal chromosomes synapse on the left-hand side of the maternal chromosomes in red. As a result, the microtubules in meiosis 1 pull both blue paternal chromosomes to the left and both red maternal chromosomes to the right. In meiosis 2, the homologs have already been separated, and so the only thing left to do is to separate the sister chromatids, resulting in 2 gametes with only blue paternal chromosomes and 2 gametes with only red maternal chromosomes. Now, look at the meiosis on the right. Just by chance, the longer blue chromosome synapses on the left, and the shorter red chromosome is also on the left. When the spindle fiber is attached to the kinetochore, they pull the larger blue chromosome and the smaller red chromosome to one cell, and the chromosomes on the right to the other cell. Again, in meiosis 2, the sister chromatids are separated, resulting in the combinations we see at the bottom. In all, there are 4 combinations possible for gametes that are produced by this individual, and each of these 4 possibilities is produced with equal likelihood. Now, this was a very simple case in which this species only has 2 pairs of homologous chromosomes. Remember that humans have 23 pairs of chromosomes, and so imagine aligning 23 pairs of chromosomes along the metaphase plate. There would be about 8.5 million different possible combinations of these chromosomes. In fact, there are many times that many as a result of the recombination of independent assortment and crossing over. As a result, no 2 gametes that are produced by meiosis are ever identical. In fact, identical twins result from a single egg and a single sperm that fertilize one another and are only divided into 2 individuals later. Fraternal twins result from 2 eggs and 2 sperm, and they are as closely related to each other as any sibling pair is. We'll look in more detail at the consequences of independent assortment and crossing over when we study Mendel's laws and mapping the location of genes on chromosomes. One interesting question that biologists have asked with regard to sexual reproduction is simply, why do so many animals have sex? Consider an organism that reproduces asexually. That is, it reproduces on its own. One individual may have 4 offspring, and each of those 4 offspring will themselves have 4 offsprings, and so on. 2 generations later, there may be 16 individuals. In a sexually reproducing species, however, it takes 2 parents to make 4 offspring. And so in that first generation, the number of offspring has only doubled compared to the number of parents. By generation 3, there are only half as many offspring as there are for the asexual species. This is known as the paradox of sex because it seems that if an asexual population and a sexual population were to coexist, the sexual population would be driven extinct by the asexual population simply because the asexual population reproduces much more efficiently. In effect, sexual reproduction produces males which are numerically useless. It's a paradox because despite the numerical advantage asexual populations seem to have, sexual reproduction is much more common than asexual reproduction. One hypothesis for why sexual reproduction is more common despite its numerical disadvantage is the purifying selection hypothesis. Imagine a mutation marked here with a star that alters the DNA and is harmful for those individuals that carry the mutation. In an asexual species, imagine that it reduces the reproductive rate in half, resulting in every individual only having 2 offspring instead of 4. The mutation gets carried on because each offspring will certainly inherit the mutation in an asexual species. In sexual species, on the other hand, only some of the individuals may inherit that mutation because of the fact that meiosis produces gametes with only 1 copy of each chromosome. Those individuals that inherit the mutation may indeed have fewer offspring and may pass the mutation on to their offspring. However, some of the offspring may not have that mutation and will maintain the same higher reproductive rate. A mutation like this eliminates the numerical advantage that an asexual species has. Evidence has been found to support this hypothesis from a tiny water flea called Daphnia. Some species reproduce sexually and others asexually, but the asexual species has many more deleterious, harmful mutations than the sexual species. The second hypothesis is known as the changing environment hypothesis. According to this hypothesis, asexually reproducing species are much more vulnerable to a changing environment because if the environment is such that one type cannot survive well, all of them will not survive well since they're all genetically identical to one another. The environment is always changing, and this change may be slow over geologic time, or it may be rapid, as may happen in the presence of new diseases. For a sexual species, each offspring is different, so some may be vulnerable to the new environment, but others may survive well. Those that survive well will produce offspring of their own that have similar genetic makeup that allow it to survive in that changed environment. Evidence to support this comes from many experiments, including ones with the nematode C. elegans. In C. elegans, there are no females, but there are males and there are hermaphrodites. Therefore, individuals may reproduce either by self-fertilization or by mating with another individual. If populations of C. elegans are grown with or without a bacterial pathogen, researchers have found that the rate of sexual reproduction goes up in the presence of the pathogen and it goes down in the absence of the pathogen. This suggests that the change environment encourages sexual reproduction and gives an advantage to those individuals that do outcross. This advantage is likely to be some resistance to the pathogen. Finally, the Deloid rotifer is an asexual species and has been for more than 50 million years. Scientists think that it has survived for so long even without sexual reproduction because of its environment. These small animals live in ponds that may dry up for long periods of time, and as the ponds dry up, So do the animals. Their membranes may crack and allow foreign DNA to enter. Sometimes this foreign DNA is incorporated into the deltoid genome. Therefore, even though these organisms don't have sexual reproduction, they do sometimes mix up their genetic architecture, and that may help them evolve and survive in changing environments.